Okay. Hi, my name is Ethan Tanchoco, and I will be presenting on the effects of a sorry. I'll be presenting on the effects of a lactobacillus-derived bacteriocin on Vibrio cholera infection and the microbiome. So first, let's establish what Vibrio cholera is. It is a bacterial and diarrheal pathogen that affects roughly 3 million people worldwide every year. And although treatments are available for this disease, there is a need for more accessible strategies that reduce the economic burden in developing countries, which are the main targets and sufferers of this disease. Vibrio cholera infection is pretty well described, but its interaction with the gut microbiome, which is the totality of viruses, bacteria, and fungi that naturally live in our gut, those interactions are not very well defined. Due to this gap in the literature, we believe that it could provide key insights on cholera treatment and on new information about gut physiology as a whole. And so, as you can see here, we have the elaborate infection cycle of Vibrio cholera. But for the purposes of this presentation, we just need to know that Vibrio cholera will release a toxin into intestinal epithelial cells. And then through a cascade of events, we result in the release of ions into the gut. And as a result, we also get water loss and water excretion into the gut, and we get the diarrheal symptoms that are characteristic of this disease. And so my work will be continuing on from the paper by Hu et al., where they found that Lactobacillus gasseri LA39 provided resistance to stress-induced diarrhea in piglets. This resistance was attributed to gassricin A, which is an antimicrobial molecule that's produced by LA39. As we can see here in this diagram, gassricin A essentially binds to a receptor on intestinal cells called KRT19, and this upregulates the fluid absorption and downregulates the fluid secretion in these intestinal cells. And the net effect is that we get a reduction in the amount of fluid accumulation in the gut, so less water and therefore less diarrheal symptoms. So for my project, the mechanism of Vibrio cholera-induced diarrhea really relies on increasing fluid secretion and decreasing fluid absorption in the intestine. We observed that both LA39 and KRT19 are found naturally in humans, and therefore also gassricin A directly opposes Vibrio cholera's mode of action. Due to these two observations, we hypothesized that L-gasseri LA39 abundance in the gut can increase resistance to Vibrio cholera-induced diarrhea by releasing gassricin A and therefore reducing the amount of water accumulation in the gut and reducing diarrheal symptoms. To test this, we created a mouse model where we start off with a cohort of mice and give them antibiotics to remove the natural microbiome of mice. And then we would split them up into two experimental groups. First, we would give JCM1131 and wild type Vibrio cholera, and also LA39 and wild type Vibrio cholera. The main difference between these two is that although they are both lactobacillus strains, LA39 will produce gassricin A, but JCM1131 does not produce it. And so we would let these incubate for a day, and then we would measure or weigh the intestine of these mice to get the fluid accumulation ratio. And what this would tell us is that higher fluid accumulation ratios indicate more water in the gut and therefore more potential diarrheal symptoms. A lower fluid accumulation would give us less water in the gut and therefore less diarrheal symptoms. The second measurement for this experiment would be to measure Vibrio cholera colonization, and that would just tell us how much Vibrio cholera is successfully colonizing the gut in infection. So preliminary results show that Vibrio cholera in the presence of a non-producing strain, so 1131, gives a higher fluid accumulation ratio, whereas Vibrio cholera in the presence of gassricin A producing lactobacillus, so LA39, causes a lower fluid accumulation ratio. Due to this difference, we believe that the presence of gassricin A could potentially reduce the water accumulation in the gut and therefore reduce fluid accumulation. In fact, the gassricin A producing strain was able to reduce fluid accumulation to levels similar to a mice that did not, or a mouse that did not have Vibrio at all, or Vibrio cholera at all, and instead a PBS control. Additionally, in terms of colonization, we found that there was a slight but significant reduction in the colonization of Vibrio cholera in the gut in the presence of gastricin A. And so further questions would be to ask whether there's a direct effect on Vibrio cholera due to gastricin A, or if this is simply due to a competition for resources between LA39 and Vibrio cholera. In terms of my project, this is kind of where it took a pause due to the pandemic. However, future plans for this experiment would be to first utilize an insertional mutagenesis technique, where we noticed that JCM1131 and LA39 have growth differences that could potentially skew the results. And as a result, we plan on inserting a erythromycin resistance gene in the middle of the gastricin A producing gene. And as a result, we would get LA39 delta GAAA and LA39. The main difference between these two strains would be that LA39 can produce gastricin A, LA39 cannot, but they are similar in every other aspect or characteristic. And this would establish a clear control for future experiments. And so as we can see here, we would have a modified experimental design where instead of using 1131, we would use LA39 delta GAAA. In terms of investigating Vibrio cholera's interactions with the microbiome, there are three main observations from previous papers in our lab to consider. First of all, the microbial communities of patients recovering from cholera were susceptible to future cholera infections. Communities from healthy patients were found to be resistant to cholera infections, and that Blauschia obium was found to be the main driver of resistance against cholera infection, whereas E. coli, Streptococcus, and Enterococcus faecalis were associated with the susceptibility to cholera infection. So we observed also that aside from its effects on KRT19, gastricin A in the literature has been shown to specifically target and inhibit some gram-positive bacteria. Blauschia obium, Streptococcus, and Enterococcus faecalis, which are all key microbiome members in infection to cholera, are all gram-positives, and this resulted in us developing two main questions to answer in the future. First of all, we wanted to ask if LA39 can outcompete negative bacteria in these susceptible microbiomes and create or turn susceptibility into resistance by inhibiting those negative bacteria, and also whether LA39 could be potentially conjugated or included in treatment with Blauschia obium to enhance the positive effects and the positive resistance against Vibrio cholera, or if there would be an anti-synergistic effect in which LA39 inhibits the growth of Blauschia obium and reverses the resistance to cholera. In terms of our experimental design, we would use a similar mouse model. However, the main difference would be that LA39, one group would be LA39 with the negative microbiome members, and the other would be LA39 with the positive microbiome members. From there, we would test the interactions through fluid accumulation ratio, Vibrio cholera colonization, and then also 16S qPCR and sequencing to tell us the abundance of these bacteria at the end of infection. So through continued work on this project, we think that gastricin A's effect on Vibrio cholera infection could be further elucidated, and current data hints and suggests that gastricin A producing LA39 may be useful in a potential human-derived probiotic against cholera. This would give us a treatment that doesn't require the intensive resource allocation that oral rehydration solution treatments require, and this would give a much more economically viable solution for developing countries against this reoccurring disease. Still, there are many unanswered questions to consider in a translational perspective, such as how gastricin A affects the severity of diarrheal symptoms, how whether it's more effective administered before or after Vibrio cholera infection, and also its effect on the overall gut microbiome structure for patients long-term. And in blue, we see the types of models we could use to answer these questions. And so, yeah, I just wanted to thank the Dr. Shao Lab, as well as the Chancellor's Research Fellowship for all the support in the project, as well as the National Institute of Health for funding the project. And so here are my references, and thank you for your attention.
Thank you for your presentation. You were also right on time. And now we're